webinar aajok Mashurul Hoque Foundation in partner with Saara Telemedicine. সঞ্চালক হিসেবে আছি আমি Shahneawz খান। শুরুতেই Mashurul Hoque Foundation সম্পর্কে একটু বলতে চাই। সেটা হচ্ছে একটি স্বেচ্ছাসেবী charity organization. Basically দীর্ঘমেয়াদী এবং টার্মিনাল ইলনেস ভুগছেন এবং তাদের স্বজন এবং তাদের শুভানুধ্যায়ীদের সাথে থাকার প্রত্যয় নিয়েই এই Mashurul Hoque ফাউন্ডেশনের পথযাত্রা। কিন্তু এর রেজিস্ট্রেশন প্রক্রিয়াধীন এখনো, কিন্তু ওয়েবিনারের মাধ্যমে তারা এখন মানুষের পাশে থাকতে যাচ্ছেন এবং এই আয়োজন হচ্ছে তাদের জন্যই। এই আয়োজন বর্তমানের কম্পিউটারি যেসব গবেষণা হচ্ছে স্বাস্থ্য খাতে এবং সেই বিষয়ে জানা এবং সবাইকে অবহিত করা, এই ব্রত নিয়েই আজকের ওয়েবিনারের আয়োজন। তো আজকে আমাদের প্রধান আলোচক Gitalee Sarker, ডক্টর Gitalee Sarker। উনি নভো নর্ডিক্স পোস্টডক্টর ফেলো এবং আমাদের এখানে সামারভিল কলেজ, ইউনিভার্সিটি অক্সফোর্ডে রিসার্চ ফেলো আছেন, হিসেবে কর্মরত আছেন। Dr. Gitalee has an outstanding research career so far. She has completed her MBBS from Dhaka Medical College, and then she has MS in neuroscience from University of Bonn, Germany. Then she took her PhD in neuroscience at ETH Zurich. বর্তমানে Dr. Gitalee the research focus হচ্ছে neuroimmunity in obesity and cardiac function. So we are very pleased to have Dr. Gitalee with us today. On behalf of Mashurul Hoque Foundation, I really thank Dr. Gitalee to be present here. আজকে ডক্টর Gitalee আমাদের obesity নিয়ে আলোকপাত করবেন। বিশেষ করে গর্ভাবস্থায় মায়ের যদি অস্বাস্থ্যকর খাদ্যাভ্যাস থাকে এবং তার এফেক্ট তার সন্তানের উপর এবং তার পরবর্তী প্রজন্মের উপরও কিভাবে একটি ইফেক্ট সৃষ্টি করতে পারে, সেই বিষয়ে একটাই গ্রাউন্ড ব্রেকিং রিসার্চ আমি বলবো, সেটি নিয়ে আমাদের সাথে কথা বলবেন এবং শেয়ার করবেন ডক্টর Gitalee Sarker। এখন ফ্লোর আমি ডক্টর Gitalee Sarker এর কাছে দিচ্ছি। প্লিজ প্রসিড অন ইউর প্রেজেন্টেশন।
Thank you, Dr. Khan. Let me first upload my slide. Okay, can you see my slides?
It's fine.
Great. First of all, I would like to thank Mashurul Hoque Foundation to give me this opportunity to present my research work. So hello everyone. I don't have to introduce myself again after this nice introduction from Dr. Khan. Today I'm going to talk about the research work that I did during my PhD in ETH Zurich. And the topic is the multi-generational effects of obesity. So I will start with a short background about this project. So I think we all know that we are in a big fat crisis. We know that obesity actually induces the risk of several non-communicable diseases. As you can see here, diabetes, cardiovascular disorders, fatty liver disease, different type of cancers, and overall also it affects our mental health. Not only that, during this COVID time, we actually found that nearly 1/3 of the severe COVID cases were blamed on obesity. So basically, obesity increases the risk of COVID-related mortality. So it's a huge problem. It's a big public health concern of the modern world, because it not only reduces our life expectancy, it also reduces our quality of life. And in this era of pandemics, I would like to say obesity is considered as a silent pandemic. Over the last 30 years, the prevalence of obesity has been tripled throughout the world. So, for instance, since 1975 until 2016, it has been shot up very fast. And if we see, actually, the risk is more in women compared to the men. So if you see the trend here, that the women are more has the more tendency to become obese compared to men. And if this if this trend continues in this way, it has been estimated that over more than 1 billion adults will be obese by 2030. So what causes obesity? Why it's happening, right? I think more or less we know the fundamental reason of being obese. So several factors are responsible for it. We don't know exact mechanism, but we know the factors. For instance, so the body weight, actually, why we gain weight, it's the result of disbalance between food intake and the energy expenditure. So let's say in the modern world, it's very easy to have access to palatable, cheesy, calorie-dense food. And that actually increases our overall calorie intake. On the other hand, if we look at our physical activity, the sedentary lifestyle and lack of exercise actually reduces our energy expenditure. So together, these factors actually create an obesogenic environment. On top of that, there is genetic factors which play a huge role to determine which individual will be more susceptible to develop obesity compared to the other. However, these genetic factors or the environmental factors, they alone cannot really explain this dramatic rise of obesity over these last few decades. And therefore, a new concept actually has developed, and that is called developmental programming. So, what is developmental programming? So, it is the process by which an environmental insult, for instance, it can be dietary insult, let's say undernutrition, overnutrition, it can be stress, it can be infection during the critical period of development. So, what is that critical period? That means during the time of pregnancy, the fetal stage, and right after birth, the lactational period or the breastfeeding time of the fetus, of the newborn. So, during this critical period, this environmental insult can induce an adaptive changes in this growing fetus that may lead to do irreversible changes in the developing organs and the physiology of the offspring. And that can ultimately show up in the adult life as a form of different diseases. For instance, obesity, type 2 diabetes, it can be anxiety, or it can be other cognitive disorders. And not only that, this impact of early life insult can be transmitted across multiple generations. So, current state of research. So, what has been done so far based on this concept of developmental programming in respect to obesity? Several human and animal studies have linked this escalating growth of obesity or overnutrition during the pregnancy time and lactation with this increased predisposition to develop obesity later in life. And actually, a handful of animal studies have shown that exposure to high fat diet, which is basically it mimics the calorie dense junk food diet in our daily life. So when we exposed a rodent to high fat diet, we could see the impact of obesity. That means the subsequent two generations being obese and being developing diabetogenic phenotypes. However, what we don't know whether it is transmitted after this second generation, that is after the immediate and the after the, let's say, children and the grandchildren, whether it's going to the great grandchildren. We don't know. We don't know what are the mechanisms behind this transmission of the phenotypes. And overall, there is very less research have been done to understand the impact on our brain and our behaviors. So, in my project, what I aimed to investigate is that whether maternal high-fat diet exposure in mice can program our feeding behaviors and the metabolic state of the immediate offspring, and what are the underlying neurobiological mechanisms. I also checked whether this impact or whether these effects are transmitting across multiple generations. And if yes, what are the underlying mechanism? So to identify the underlying mechanism, I focused on the epigenetic markers. Not only that, I also investigated whether this maternal high-fat diet exposure can cause cognitive impairment across generations. And last but not least, I also checked which specific period, which critical windows of development are more vulnerable for this high-fat diet insult. So the findings of my of these studies actually gained. These findings were well accepted, in the scientific community, and it got wide media coverage across the world. But for the interest of time, I actually am going to show some of these interesting observations, and so I will focus on the first three studies. So in the first study, what we checked is the effects of maternal overnutrition or obesity on immediate offspring. Well, my model was mouse model because, of course, I want to explore or investigate the underlying mechanisms, and that's why I need a model like that. So what I did, so I put this female mouse on high-fat diet. So the high-fat diet, as I said, it mimics the normal junk food that we eat in McDonald's, let's say. So this contains 60% of calorie coming from the fat. So I put those mice on high-fat diet for 9 weeks, which means 3 weeks before conception, 3 weeks during the pregnancy time, and 3 weeks during lactation. So this is basically the overall gestational phase of a mouse, total 9 weeks. And then when the offspring were born and they were able to eat by themselves, let's say the starting of weaning, at that point of time, the offspring were on normal chow diet, which has really very low percentage of fat. So that means they were only exposed to high fat diet when they were inside their mother's womb and when they had the breastfeeding time. Apart from that, they didn't get any high fat diet unless I provided them. So what I found, so I checked their obesogenic phenotype. I found that these offspring, the immediate offspring born to these high fat diet exposed mothers, they actually gained significantly more weight compared to the control group. So what is that control group? So the control group is basically, I took another cohort of female mice, which those mothers were only fed on normal chow diet, and I checked their offsprings for the same in the same way. So compared to the control group, I could see that these mice, these offspring born to the high fat diet mothers, they gained more weight. Not only that, they had higher fat content in their body, they had higher insulin level, and they had higher cholesterol level, free fatty acid, and triglyceride in their blood. So they clearly show obesogenic phenotype. Then I also checked their behavior because I wanted to know what's the reason of gaining this weight. So to understand the reason of gaining weight, there is two ways we can check. One is their activity, and the second is their food consumption, right? So to check the food consumption, I found that nor in their day-to-day life, they don't show any difference in their food intake. However, when you give them high fat food or high sugar content food, you can see that these offsprings prefer to eat more this sucrose or the fat content food. So they have this preference for this sugary, calorie dense food. Not only that, they also showed addiction-like phenotype to different kind of addictive substances. For instance, if you gave them alcohol, which is basically instead of alcohol, I gave them ethanol, or you put them on cocaine or amphetamine, you could see that they have more tendency, more sensitivity for alcohol consumption or amphetamine or cocaine. So they have a phenotype of drug addiction. So that's very interesting to see, like they not only develop the food addiction, but they also develop addiction to different addictive drugs. So I want to know what is the underlying biology. So to understand that, well, I checked for what are the system that has been shared between food and drug addiction. And we can see here that there are subtle differences between addictive eating and classic addiction, but there are some shared system. And the biology behind that, behind these two systems is basically the brain reward circuitry, particularly the dopaminergic circuitry. So this is the mouse brain in a sagittal section. And you can see here the ventral tegmental area, nucleus accumbens, and dorsal striatum. These are the main regions responsible for controlling our liking behavior. So I checked the dopaminergic system, that means the protein dopamine, the enzyme that produces dopamine, that is tyrosine hydroxylase, in this region. And interestingly, I found in these offspring that are born to high fat diet exposed mothers, they actually have low level of tyrosine hydroxylase, which is the key enzyme for the production of dopamine. And of course, if I see less tyrosine hydroxylase, I could expect to see lower level of dopamine in all these regions. So that actually made me to conclude that maternal high-fat diet during this pregnancy and the lactation time induces a hypodopaminergic state that actually leads them to eat more high-fat content food and also to develop obesity. So after these observations in the first generation, my next question was, are these effects transmitted in the subsequent generations? To answer that question, we designed a big study. So we developed or generated mice of 3 generations. So how did we do that? So we followed the paternal lineage. What does that mean? So what happens? We first generation. So the F0 is the mother. So we have 2 groups, control and high fat diet. Control groups, mainly the mothers, that is the F0. They received 9 weeks chow diet. And the high fat diet group, here the mothers receive 9 weeks high fat diet. Then their child born. So we took the male offspring in both groups. So what we did, the male offspring were then crossed with a naive female offspring, female mice, not offspring, the female mice. So we just took wild type female mice and we crossed them with this F1 offspring from the control group or the F1 offspring from the high fat diet group. And that led us to generate the F2 offspring, that is second generation offspring. So now the second generation offspring, we again took the males and then we crossed them with wild type female mice in both groups. And in this way, we developed the third generation offspring F3 from both the control and the high fat diet group. So that actually follows the paternal lineage because we always took the male offspring in all groups. And here I would like to mention that the high fat diet insult or the food insult that only happened in this F0 generation, that means in these uh, mothers in the F0, which is the great grandmother of the F3 generation. And the rest of the time, all of them actually received the normal chow diet. So that's the study design. So now what happened? So we know until F1 what we have seen, what I have just showed you, that they have these offspring from the high fat diet exposed mothers. The F1 actually developed obesogenic phenotype. They had higher preference to palatable foods, and they also developed addiction to drugs. So now we want to see whether these effects are transmitting into the second and the third generation. So we did the similar kind of experiments. And as you see here in the left side, I did this alcohol preference test. So I provided these mice, male and female offspring from the F2 generation. So both of them, so here I have like 4 groups, female control versus high fat diet, which means female offspring born to the high fat diet exposed grandmothers, female offspring born to the chow exposed grandmothers, male high fat diet, male control in the same way. So all of them were exposed to different concentrations of ethanol. And you can see these offspring, female and male, born to the high fat diet exposed grandmothers, they actually consumed more alcohol compared to the control group, which is obvious both in females as well in the males. Next, I checked their amphetamine sensitivity. So amphetamine is a recreational drug, and it actually induces addiction in human. In case of mice, how we check that? So the sensitivity we checked in mice by their hyperactive response. So when you inject amphetamine, the mice starts to so as you see here, so this is the females, control is the gray, and the high fat diet exposed group is the orange, and this is the males. And you can see that in the baseline where they didn't get any injections, or in the saline injected condition, we don't see any difference between these two groups. But when we exposed them or injected amphetamine, overall, both groups shows increased locomotor activity or increased movement, but we could see that the increased movement is significantly more in the high fat exposed grandmothers, higher mothers. Similarly, we see it in the male offspring. So they clearly shows a higher sensitivity to amphetamine. In the males. On the other hand, the addictive phenotype that also transmitted up to the third generation, but we could see in the third generation, it is prominent only in the females. We wanted to know whether this dopaminergic system is responsible for this transmission of for these phenotypes. And we checked again their TH level and the dopamine level in both F2 and F3 offspring in these dopaminergic regions. And we could see that in the second generation, both the male and female shows lower TH level and lower dopamine level in all these regions. However, in the third generation, the effect is mainly present in the females. So we can say that maternal high-fat diet induced this hypodopaminergic state that we observed in the immediate offspring. It is conserved up to the third generation. So now the big question, how these effects are being transmitted across generations. So to answer that question, we decided to look at the epigenetic markers. So what is epigenetic changes? So it's basically the changes that happen in gene expression, whether a gene will be activated or inactivated. It depends on two things. One is like it can be this expression, changes in gene expression can depend on changes in the DNA sequence itself or changes in the other associated proteins, be that histone proteins that is associated with the DNA or the methyl groups. So when it happens, only the changes in this histone or the methyl group or the non-coding RNAs without doing any change in the DNA sequence, then it's called the epigenetic changes. And so far, the three persistent epigenetic markers that we know are the DNA methylation, histone modification, and the small non-coding RNAs. So we decided to look at those epigenetic markers, whether we see any changes due to this high-fat diet insult in these markers. So we started with checking the DNA methylation because that has been widely studied across different models. And not only that, DNA methylation is actually 80 to 90% is conserved in the sperm. And so we checked the DNA methylation. So we collected the sperm from this F1 male offspring because we generated the subsequent generations from this male offspring. So we took the sperm from the F1 male offspring from both the control and high-fat diet group, and also we took the sperm from the F2 male offspring from both groups. So then we checked, we did this DNA methylation assay, and what we found in the first generation, that is the F1 male offspring sperm, we could see that we found 527 total differential methylated regions between this chow diet and the high fat diet group. Here, again, I want to emphasize the high fat diet group means those who are exposed to the high fat diet during their fetal period or during the normal diet, during the lactation, sorry. So here we saw 527 DMRs, and then we go to the second generation. And here we also found 543 DMRs between the control and the However, when we wanted to see how many of them actually conserved or transmitted from the first generation to the second generation, we only found an overlap of only 6 DMRs. And it's a very little number to explain, and it's very hard to explain this persistent of such strong phenotype across 3 generations by these 6 DMRs. So that actually led us to conclude that probably sperm CpG methylation or DNA methylation may not be the major carrier for the transmission of the maternal high fat diet induced altered traits that we observed. So after that, we decided to look other epigenetic markers that I just mentioned in the last slide. And we wanted to focus on this one, the sperm small non-coding RNAs, because during the time I was doing this study, there are two a paper came out at the same time, who actually, these two papers basically looked at two different models. So one is paternal high fat diet model, and the other one is low protein diet model. So, and in both cases, they actually found changes in the sperm small non-coding RNAs. So it seems like the sperm non-coding RNAs might be a potential vector for the transmission of this kind of environmental induced phenotypes. So we focused on that. For that, so this is the third study that I did during my PhD. And here, what we did, so same way, we had these mothers, we put them on high fat diet or normal chow diet. Then we developed this first generation offspring from both groups. From the males, because we focused on the paternal lineage, so we took the male sperm and we isolated the sperm, and from the sperm we actually extracted the total RNA. And then what we did sequencing of this sperm total RNA from both high fat diet group and the control group. When we did, we found that tsRNA, it is called tRNA derived small RNA, it's one kind of small non-coding RNAs. Their level is actually significantly high in the high fat diet exposed group compared to the control group. So we found our one of our candidate small non-coding RNA. So then what we did, we wanted to generate a mouse only transmitting this defective small non-coding RNA, that is the tsRNA. So for that, what we did, we again collected the total RNA from these two groups. One is the male offspring born to high fat diet exposed mothers, and one is the male offspring born to normal chow fed mothers. Okay. So what we did here, then we actually, by gel electrophoresis, we actually separated different RNAs, the small non-coding RNA. So we found tRNA, we found tsRNA, and we found another band, which is we couldn't really explain what is that specific type of specific type of small non-coding RNA. However, we focused on this band, which is not very clear here, but there are like 4 bands present at the level of 30 to 40. So these bands are the tsRNA bands. So we then extracted this tsRNA from these gel bands, purified them, and then we injected this tsRNA into a normally fertilized oocyte. And then we developed this zygote. This is kind of the same technique that used for in vitro fertilization or IVF. So in a petri dish, we basically put this tsRNA into this fertilized oocyte. And then once this zygote is developed up to a certain stage, then we transferred this zygote into pseudo-pregnant female mice. So in this way, we actually generated a high-fat diet tsRNA group and a control tsRNA group. So uh, uh, cool technique, we actually were able to get this tsRNA offspring from the high-fat diet exposed mothers and the control exposed mothers. So then what we did, we checked these behaviors, the feeding behaviors, their preference to high-fat diet or sucrose in the similar way that we did before. And we can see both in females and males. So we checked both sexes, and we can see that the male high-fat diet tsRNA offspring showed higher preference to high-fat diet compared to the control. Similarly, the sucrose preference was found in the high-fat diet male tsRNA offspring compared to the control. We then checked their addiction to drugs. So we put them on alcohol preference or amphetamine sensitivity, and we could see that both male and female could show the preference to alcohol or the amphetamine, the one from the high-fat diet tsRNA group. So that actually confirms that high-fat diet tsRNA offspring conserve the enhanced hedonic response to palatable foods and drugs of abuse, similar to their ancestors. Next, we check their obesogenic phenotype. And as you see here, similar to their ancestors, both the females and the males actually gained weight, although the weight gain is more prominent in the male high-fat diet tsRNA offspring. They have also showed increased fat content in their body. They showed impaired sensitivity to insulin, and they showed higher cholesterol level. And that's more prominent in the male high-fat diet tsRNA offspring compared to the other groups. So that also confirms that high-fat diet tsRNA offspring developed this strong metabolic phenotype following a free palatable food choice test. So if I now summarize these three studies, what I can say that what we observed when we put those mice on 9 weeks high-fat diet, 3 weeks during preconception, 3 weeks during gestation, and 3 weeks during lactation, the immediate offspring, the first generation, they gained weight, but that was mild obesogenic traits, which was observed both male and females. They also developed addictive-like behaviors, and that was partially explained by their hypodopaminergic state in the reward system. And these three traits were all conserved in the second and also in the third generation. So it shows a multi-generational effect. However, in the third generation, we saw a sexual dimorphism where the females showed more addictive-like behaviors and the male showed strong obesogenic traits. When we looked for the mechanism for this transmission, we found that sperm DNA methylation couldn't explain. However, sperm tsRNA transmission could reproduce this phenotype in the injected offspring. So what's the impact of this study? So this study, I mean, first of all, it actually emphasizes the importance of healthy food habits during pregnancy and lactation. And another thing, it also identifies sperm tsRNA as a essential prognostic biomarkers of obesity that can be used in future in sperm quality assessment during IVF. However, to implement it as a prognostic marker, before that we have to answer a lot of other questions. So what are those questions? First of all, we want to understand the mechanism by which sperm tsRNA actually mediate these altered traits. Second of all, we want to explore how these tsRNA mechanisms can be reversed to prevent obesity across these generations. And third, to develop new technology to sort out healthy sperms based on this tsRNA quality assessment. So with that, I would like to with that, so are we still in live?
Yeah, you are still
Okay.
live. Thank you, Dr. Gitalee. It was really nice presentation and really nice information to pass on to the next generation, to our competitive people, and also the mothers who are going to be mothers in the next in future. So যারা সত্যিকার অর্থেই মায়ের খাদ্যাভ্যাস কিভাবে আমাদের জেনারেশনকে নেক্সট জেনারেশনকেও এফেক্ট করতে পারে সেই জিনিসটা জানা এবং আমাদের খাদ্যাভ্যাসের প্রতি আমাদের যত্নবান হওয়াটাই আসলে এখানে এমফাসিস দেওয়ার বিষয়। আপনার অসাধারণ কাজের জন্য আমি জানতে পারলাম, যদিও মাউস মডেল, বাট উই হ্যাভ দ্য বেস্ট মডেল টু হ্যাভ। এছাড়া আমাদের কোনো মডেল নেই, সুতরাং যে অবজারভেশন আপনি দেখিয়েছেন well accepted in the scientific community, and also really good information for the people in general. তো আমি আবারও মশরুল হক ফাউন্ডেশনের পক্ষ থেকে ডক্টর গীতালি সরকার আপনাকে ধন্যবাদ জানাচ্ছি আপনার প্রেজেন্টেশনের জন্য। আমাদের নেক্সট ওয়েবিনার নেক্সট স্যাটারডে 8 PM, আরেকটি ইন্টারেস্টিং টাইটেল, ট্রেনিং উইথাউট ট্রেনার্স, ফিউচার অফ স্কিল ডেভেলপমেন্ট অপরচুনিটি। সো সেই পর্যন্ত আপনাদের আবারও ধন্যবাদ জানিয়ে আমাদের নেক্সট ওয়েবিনারে আপনাদের অংশগ্রহণের আশাবাদ ব্যক্ত করে আমরা এখন ওয়েবিনারটি ঘোষণা করতে যাচ্ছি। ধন্যবাদ।